Good afternoon, everyone. So welcome to our very last CCOVI Lecture of the lecture series for 2017. This afternoon, it's my pleasure to introduce my colleague, my friend, Dr. Tom Lowery. Tom is joining us from Agriculture and Agri-Food Canada in Summerland at the Summerland Research and Development Centre. But Tom also spends some of his time here in Ontario at the Vineland Research and Development Centre for Ag Canada as well. Tom also is one of our CCOVI professional affiliates, so one of our members of our research team in CCOVI. And he's also affiliated with the University of British Columbia in the Okanagan Valley in Kelowna, BC. He is also the 2013 recipient of the prestigious Queen Elizabeth II Diamond Jubilee Medal. So congratulations on that, Tom. I didn't know that. He has also served on a number of BC Wine Grape Council research and development committees, so he's very active in assisting the BC industry in a lot of their programming, and has produced the Insect and Mite chapter of the BC Production Guide for Grapes, with accompanying photo guides for grapevine pests and beneficial insects. So a lot of grape growers rely on that sort of guide as they're managing their vineyards. For the past 20 years, Tom has been conducting research to develop a sustainable grape pest management program for British Columbia. The broad scope of his integrated pest management research includes many non-chemical methods, as well as chemical and biological controls, leafhopper anti-feedants and repellents, and the use of beneficial vineyard ground cover vegetation for the management of grapevine pests. Additionally, his work on the epidemiology and management of virus infections of woody perennial plants includes research on grapevine viruses and the operation of a lab at the AAFC Vineland station, where he studies plum pox virus on stone fruits. So today he's going to talk to us a lot about the work that he's been doing on grapevine viruses in British Columbia, and we're all actively here to learn because there's a lot of application for issues that we're struggling with here in Ontario, as well as across the country in our other grape and wine producing regions in Quebec and Nova Scotia. So with that, Tom, I'll leave it to you.
Thank you, Deb. I've rolled up my sleeves. Time to get down to business. Thank you very much. And first, I want to thank Deb and the organizers, Sarah, of the lecture series for allowing me to come at this time and combine it with some other work. I know it's a little late in the lecture series and most of the students are away, but I know there'll be industry people and others online that hopefully will get a lot of information from this. So I'm going to be talking about the epidemiology and management of grapevine viruses. Before I do, I want to give a lot of credit to our postdoctoral fellow on this project, Dr. Sudarshana Poojari. Sud's been with us for the last 3 years working on this project. And you can see from this, he's very knowledgeable, and he's one of these rare people. He does a lot of lab-based molecular work, a lot of sequencing and various lab components, but he's also great going out to the field, meeting with growers and doing that work. So we were very fortunate to connect with Sud. And as you can see, this is the last year and a bit. He's been extremely productive. So I really want to make sure I acknowledge Sud's efforts on this project. So I'm going to give a presentation outline. A bit of background in the current status of the virus diseases mainly in BC. It's a bit of information for Ontario. Looking at primary virus infections and then secondary spread by insect vectors. And of importance, of course, is then what do we do about it? What's our management strategy? And then I'll wrap that all up in a summary. So first of all, just to give you some background here, there's a picture and you'll get used to seeing a lot of these. One of the main ones we're dealing with, there's grapevine leaf roll associated virus of Merlot, and we're going to get back into some details of what it looks like, what damage do they cause, etc. I want to mention here that I'm the third of the series here to come out for the lecture series, so I'm not going to go on into a lot of background because hopefully you've all heard Sud was here in March and he gave details about the development of the diagnostics, some of the work he's been doing in support of our project. Jose was here last year in March and giving an overview of the whole situation, what was the incidence of virus and vectors and everything. So I'm building on this. I also want to point out some other people who are involved closely in the projects. Dr. Pat Bowen of our center does a lot of work. So she does the Grapevine physiology. She's a very good viticulturist, works on a number of things. Carl Bogdanoff, he works on mainly right now winter cold hardiness work, wine quality, etc. He does a lot of different things. And Kevin Ushers, our chemist, works on aromatic and flavor components, what influence these virus diseases have on the fruit quality. So the other thing I want to do is, of course, thank Agriculture and Agri-Food Canada, but mainly the BC Wine Grape Council. They've been very supportive of our work. This is one of the current, we're finishing up the last year of an AIP. BC Wine Grape Council is funded investigation of grapevine leaf roll associated virus, and we're looking at a range of things, as you can see from these 4 objectives. The 2 that I'm going to deal with, mainly the identification, abundance, and control of potential leaf roll insect vectors, and then looking at the efficiency and sort of what can we do about this. And another project that we had funded through AFC was an A-based project, and this one was looking at the lesser known grapevine viruses. So we had a big one on leaf roll, it's the major concern, but we've also looked at fan leaf and a whole bunch of other things. So I was the PI on this one, and I'm mainly focused on identification, abundance, and control of the insect vectors in British Columbia. So as some background, so grapevines are known to host more infectious agents, viruses, viroids, and phytoplasmas than any other woody plant species, and in fact, probably any other plant species. And there's a number of reasons for this. They're long-lived perennial plants. Most of the production of new plants is through vegetative propagation. A lot of viruses aren't seed-borne, so if you're not reproducing by seed, you're more likely to keep these in the mix. We also mix and match a lot of materials. So we take rootstocks and scions and various other ways that we mix them in. They're grown in many regions of the world. So if there's a disease in one part of the world, there's a good chance it's going to be picked up and the plants get moved around. There's a long production history. Some records say it goes back to 7,000 BC in China, in Europe even, 5,400 BC. So there's a long history. There's a lot of time there for virus infections to get into the material. Nursery stock is easily stored and transported, so you can move it all over the world. Again, anything that pops up in any part of the world is probably going to end up everywhere. We also have a mix of species. We have wine and table grapes, and there are also, don't forget, a number of wild vitis species. Some of those have gone into the production of rootstocks, for example, but that's another way that viruses or diseases can be picked up. Also, many major pests of grapes are also important virus vectors. I'm going to go through a couple of things. Here's just a graphic representation of the ways that the virus can get into your nursery material. So we've got down here, we've got the opportunity for a virus-carrying vector to introduce virus to your mother plants. So sometimes they don't show symptoms right away. There can be quite a latent period. So it can get into your mother plants and you can propagate from that, either cuttings for more mother plants, or if you use that scion material and there's virus in it. And of course, also, they can infect the rootstock material. So when you're combining these two, it can come in through the rootstock, through the scion, and you can end up propagating these virus-infected material. So the other thing I mentioned, they're very easy to propagate relative to a lot of other plants vegetatively. So you can take cuttings, or you can graft them, you can move them around the world. They package well, store very well, so you can hold them for months, you can ship them all over the place, and they'll survive quite nicely. So this material for BC, we bring in material, some from Washington State, some from California, some directly from France, rootstocks from Germany perhaps, or maybe it comes to Ontario first and then to BC. So this is just a partial picture. Material is moving all around. There's also local production. Some people own rooted material, so they're taking cuttings, maybe not being too careful where they're getting it. You go out in the winter, you can't tell if it's infected, you're getting that material. You might be just own rooted, or you might be using it for sign material. Your neighbor has nice grapes, so you want to use those. So if you're top working too, some people do that. They're converting them over. So all these various ways can lead to greater amounts of infection. I mentioned the Hemiptera. This is a suborder of the Hemiptera. So these are insects with piercing, sucking mouth parts. And we have a large number of insects of this group, this subfamily, suborder I should say, that are major pests of grapevines. So throughout the world, wherever you grow grapes, these are some of the most important pests that you have. And these include aphids, grapevine aphid, that occurs here. Grape phylloxera occurs here. You all know about that one. Leafhoppers, we have a number of species. Tree hoppers, buffalo and three-cornered alfalfa tree hoppers. I'm going to come back to why they're important in a little bit. Sharpshooters, mealybugs, great mealybug here, but in other parts of the world there's a whole slew of them. California has a big problem because they have vine mealybug and a lot of other species. Soft scale are important. We have a number of species here in Canada. And even a psylla, the hot hornbeam psyllid is a problem, a pest problem. So here's a picture of the great mealybug from BC. You have it here. Here's the cottony maple scale. You have that here. So we are dealing with, as pests of grapevines, we're dealing with a number of very efficient vectors of grapevine viruses. So we have the viruses and we have the vectors, so we know there's a lot of virus around the world. Wherever you grow grapevines, viruses are a major challenge. We had very little information concerning the virus status of vineyards in Canada. The previous survey was done in 1994-95 by the CFIA. They surveyed vineyards across Canada, and here are the results. They looked at the major ones at that time that at this time were under regulation, international regulation. So they were regulated diseases. So they went and looked and found most of them vineyards right across Canada. So grapevine leaf roll associated virus 3, you can see it occurred everywhere they looked. It was a relatively low rate, and I've highlighted Ontario here. You see there was actually quite a bit more in Ontario than anywhere else. And then also grapevine leaf roll associated virus 1, and everywhere except Quebec, and fanleaf and Arabis mosaic virus. So they found all these. Because they found them across Canada, and because of some pushback by industry members and others, they deregulated all these. So as of the mid to late 1990s, these were deregulated. So we started in 2011 conducting surveys. We needed to find out in BC which viruses we had, which ones were most important, where were they located. So we looked at all the major sampled in intensive sampling throughout the different regions or production areas in the Okanagan Valley and the Similkameen Valley that runs sort of parallel to it. Also sampled down, there's some great production on Vancouver Island and in the Lower Mainland. So we used a protocol. It was a modification of Fuchs' system of sampling. So Fuchs works in New York State, and he did a lot of work there showing the incidence of virus. And so what we did, we sampled 128 commercial vineyard blocks, and in, as I mentioned, the 6 great growing regions in BC. So we pretty well did the whole grape production throughout British Columbia. The symptoms, the titer is highest in the fall, so that's where we went August and September. We did a quadrant sampling where you collect 4 leaves from 5 consecutive vines per panel, and those get lumped into one sample. And then we did 20 of those samples in a vineyard block. So you're sampling actually a large number of vines with this. So a single vineyard block is equivalent to sampling 100 vines. We didn't prioritize, we didn't look for symptoms, and we didn't avoid vines with symptoms. I should mention, in addition to this random sampling, we did do targeted sampling because we were interested, you know, if we found something that looked unusual, of course, so hundreds of targeted samples as well. So, random samples collected from, we ended up sampling 3,261 panels of 5 vines per panel. 241 cultivar blocks eventually showed little change in the incidence of grapevine family virus or Arabis mosaic virus. So these were ones that they detected in 1994-95. They had not changed at all. Not too surprising for family virus. The known vector, a nematode, we aren't known to have it. So we were pleased to find out the incidence hadn't changed at all. What had changed was the levels of grapevine leaf roll-associated viruses. Those were much higher in our tests, and I'll get to that in a minute. And also, this was the first records for grapevine red blotch virus in British Columbia. So I'm going to touch on that in a little bit, but I wanted to point out here the symptoms. This is a typical leaf with grapevine leaf roll-associated virus. And the two things that really set it apart, the interveinal reddening of the red varieties while the veins remain quite green, and also a downward curling of the margins of the leaves. Symptoms are fairly similar for red blotch, and they can be confused when it's early onset of the virus. The symptoms are mild and they'll both sort of show a bit of red blotch. But there are a couple of differences. One is that these often get a bit thickened and coarse, and they curl up sometimes. You can see a little bit of it there. As it progresses, you get some of the veins become reddish. So the veins do go red versus they stay quite green for leaf roll. So there are differences, and you can see here in a lot of them, they do tend to have more of a pinkish red than this dark, nice dark red. But a lot of times you can be fooled. You have to do the testing. You need to do the SUD does a lot of work verifying, doing sequencing, and various types of testing to verify this. So here's what we found, and it's important here to show differences, and I'm going to come back to this later. We sampled throughout the North Okanagan, the Central Okanagan, the South Okanagan, Similkameen, and what I want to point out from, if you remember earlier on, 2.15% leaf roll virus 3 and 1.28% leaf roll virus 1 in 1995, and you can look, for example, down here in the South Okanagan, over half of and these were random tests, over half of them were positive for leaf roll virus. So there's an awful lot of it. So why are we concerned? Grapevine leaf roll disease, it's the complex of leaf roll viruses. They cause significant damage to the grapevines. The viruses colonize and reproduce in the phloem, the phloem tissue, disrupting the flow of nutrients to the shoots, leaves, and fruit. So they're stressing the vines, they're reducing the buildup of carbohydrates, and this has a negative impact on fruit quality characteristics. So you get decreased berry weight, as you can see up in here. There's a typical, from a healthy cluster, and there's from a diseased vine, you get less fruit on the clusters, smaller fruit, and delayed fruit maturity is a big problem. We're in a cool climate. We need some years all the growing degree days we can get, and this can, in some reports, delay maturity up to 2 weeks. So reduced soluble solids, higher pH, lower titratable acidity. So there's a yield reduction, but there's also a quality reduction as well. Something important to point out, infected vines cannot be cured. There's no way to get the virus out of these. No one has really shown that there's anything you can do. You can't put on more fertilizer or anything. Carl Bogdanoff is doing some work. We're hoping we can maybe find some way to get these to struggle on a little bit longer and perform a little bit better. But generally, you get this virus, those vines are doomed to keep decreasing over time because of the stress. I just wanted to show you a few pictures again. Now, this is really severe conditions. So here's a Merlot leaf. You can see the downward curling at the edges, and even with this severe case, the veins remain quite a green color. And here it is on the entire vine, and that late in the season, that's quite a stress. The photosynthetic activity is greatly reduced on a vine like this compared to a healthy vine. The green varieties, the white varieties like Chardonnay here, sometimes they show quite clear symptoms, a little bit of yellowing and the distinct cupping of the leaves. But some varieties you don't see it, and so the white varieties are very difficult. The symptoms aren't expressed clearly in quite a few of the white varieties. Moving on to grapevine red blotch virus. So it's a new virus. It was only identified in 2007 in California and really wasn't characterized until 2012. So 2012, we knew what it was. Since then, it's been shown to occur in various places throughout California, found in table grapes. You can see Maryland, Oregon, Pennsylvania. It just keeps expanding. It was also found, though, that it was isolated from a material kept in specimens that go back, I have to get the date here, 1940. They found out that some preserved specimens, that it was actually in there. So this is a virus. It's actually been around a long time, and we've just kind of overlooked it. It was probably confused with leaf roll or some other viruses to begin with. Ontario, British Columbia, etc., as I show here, Poojari et al., 2016, we reported it from Ontario and BC. In 2016, also some lab work showing that a tree hopper might be a vector. That was all lab work, but they did show transmission. And then 2017, they're doing the work on the effects on the grape fruitfulness and vine growth. So it's brand new, we don't know a lot about it, so we set out to do a survey to find out how much grapevine red blotch virus we have, how big a problem is it. 2014, we mainly sampled older vines, greater than 10 years of age. We sampled 52 vineyard blocks with, again, these random composite samples, and it all has to be done in PCR. You can't do ELISA on this one. So we only found, from all of these blocks, we ended up with 3 positive samples from 1 individual block. So things look pretty good for BC. We thought, well, this isn't so bad. We don't seem to have very much of it. 2015, we expanded it. We targeted younger vines. The problem, it really expanded in California in recent years, and we do get some material shipped up from some nursery material from California. So we focused on the younger vines. And sure enough, we did find more. So this shows all the different cultivars, whites and reds, expanded the numbers, 76 vineyard blocks, younger vines. And here we had 29 positive samples from 5 different vineyard blocks. So we do have some, but fortunately it doesn't seem to be the same extent that they've reported from California and elsewhere. Grapevine red blotch virus, I just wanted to put this picture in. You saw the symptoms on the leaves. This vine in the middle is positive for red blotch, and you can see an overall yellowing and quite a bit. It's more stunted than the neighboring vines that are free of the virus. So it does have quite a dramatic effect on vine growth and on fruit quality. And as I mentioned, Kevin Usher and Pat Carl are doing some of the work on the effects on the actual fruit quality as well. So grapevine leaf roll disease and grapevine red blotch virus, they cause similar symptoms and damage, but there are important differences, particularly when you come to the epidemiology and management. So grapevine leaf roll disease consists of several unrelated RNA viruses. So I'm not going to go into this in a lot of detail, except to say the ones we're most concerned about are leaf roll 1, 3, these ones not so much, but they're ampelloviruses. They're still within the cluster of Viridae family, but they're different members within that. This group, they're transmitted by mealybug and soft scale vectors. Leaf roll 2, there's no known vector. Nobody knows what vector, if any, is transmitting that. So that's important to know. If you have that, it's probably not going anywhere, and you can get rid of that. You don't have to be as concerned. Leaf roll 7 is interesting because it's actually transmitted what they found so far, a parasitic daughter, a plant that infects the vines, can transmit it. So it can go into the daughter, and then the daughter can infect the parasitic on the grapes, and it can transmit it. So they've shown that. In contrast, grapevine red blotch virus, it's a Gemini virus. It's a DNA virus, not an RNA virus. So different testing protocols. It's vectored by phloem-feeding homopterans again. So again, those homopterans, including the pests that we have, are important. But this time, not by mealybug and soft scale, but the Gemini viruses are transmitted more by whiteflies, leafhoppers, tree hoppers, etc. So we have to treat them separately. They look quite a bit alike. The damage is the same, but as far as the epidemiology and management, they differ. So I'm going to go into now the epidemiology. How do the viruses get into your vineyard? And we're looking at primary infection. So introduction of grapevine viruses into a vineyard occurs through various means. Planting of infected nursery material is number one. So you buy material. It has infection for some reason. As I mentioned to you, some of the major ones we're worried about were deregulated in after the mid-1990s. So there wasn't a requirement to even report these earlier on. There's greater emphasis on that now, and I think the nurseries are doing a pretty good job. But as I mentioned, you can have a vine infected. It might not show up clearly for a couple of years. Infected interplants. Sometimes you'll have a nice clean vineyard and then some die for whatever reason, and you go buy some other ones. You might only put in a few, but they might have be infected. Infected scions when top working. Movement of infected leaves and vectors on equipment. So if you go through at harvest, or if you're doing leaf removal with machinery, you've all seen it. You get leaves and debris and everything. You really should try to clean that up before you move it. We consider long-distance movement of infectious or viruliferaous vectors to be primary. So the odd one gets carried a long distance and settles in your vineyard and transmits the virus to your vines. And then vector species are responsible for secondary spread throughout the vineyard. So it comes in with primary infection, and then your vectors, your local vectors, spread it around. So how do we prevent the primary infections? Well, certified virus-free planting material. So that's number one. Try to get the cleanest material you can get. Leaf rolls and other grapevine viruses were deregulated after 1995. But testing and certification of nursery material is important. There is progress being made toward a national clean plant program for grapes. So this would be really important if we could have a strong program to make sure that the material you're putting into the ground is clean, free of these viruses. Roguing, inspection and removal of infected vines, can work in some cases, and I'm going to come back to this in a little bit and give you a little more detail. Sometimes it doesn't work, but if you have, for example, you put a few interplants in, go inspect them, have a look and make sure, because we've had lots of cases where that has happened. Nice clean vineyard, buy a few, put them in, and they're infected, and then it's just going to spread to the rest of your vines. So inspect what you've put in the ground, and if it looks like, have it tested preferably and get it out of there. Restrict the movement of equipment and workers. So I picked up this, I wanted to, just an example of this, and happened to almost immediately find this nice picture. I'm not endorsing or blaming this company that, here's a harvester going through, but I did want to point out, I don't know if you can see it there, but there's some nice red vines right in here, and I'm quite certain that's leaf roll virus. So you're going through, and it can happen early in the season to try to put your equipment into the clean blocks first, the clean areas. Just be careful, maybe wash your equipment off before you move into another site. So incidence of grapevine leaf roll viruses in BC. So how do we prevent it? I mentioned roguing. Roguing of infected vines or interplants will work in the North Okanagan, where the incidence of grapevine leaf roll is low and there are a few vectors. So here's the results from the North Okanagan. And you can see that there's really very little virus up here. Coincidentally, there's almost no vectors in this area. So if you have virus come in your planting material, roguing in the North Okanagan will work well. You can take those vines out, and you probably keep on top of this and eliminate the virus from your vineyard. In contrast to South Okanagan, for a number of reasons, I'll go into a couple of them, you have more than half of the samples tested positive. So there's a lot of virus. Unfortunately, there's also a lot of vectors. So here's a picture of cottony maple scale. It's a vector of leaf roll virus. There's a lot in the South Okanagan. That's our main production area. So you have contiguous production. There's more pests, so there's more insecticides that are required. Also, I don't know if you can see this picture, but there's little ants tending these. Ants farm mealybug and soft scale. They collect the honeydew. They also protect them from parasites and predators, and they move them around to new plants. So they have lots of the honeydew that they produce. So why is there more ants in the South Okanagan? It's quite sandy down there. So we see more species of ants and higher populations. And so that is one of the reasons, but there are several other reasons as well. So this area, South Okanagan, it's not going to work very well. So what we need to consider there is a coordinated area-wide approach that will require removal of entire blocks or even small vineyards, and establishment of buffer zones in those heavily infested areas where the incidence is high and spread is quite rapid. So this sort of situation where 100% infection, you've got this block, it's infected, there's probably lots of vectors there. If you plant a new block, a replacement block right beside it, it's going to get infected very quickly. We've had examples in our own work where a test plot, research plot was established next to a heavily infected block, and within 2 or 3 years, it was about 43% infection of the new block. So very quickly, it just blew through there. So in those areas, removal of individual blocks, roguing, that sort of thing, it's just not going to work. So what do we need? An area-wide management program where you start off. It requires, there are challenges. It requires the cooperation of many producers. So that's a difficulty in itself. Of course, it's a valuable crop, so they're not going to want to take this economic hit. So there's temporary loss of revenue. We don't know yet how wide the buffer zone has to be. We're actually working on that right now to find out how far the vectors are spreading it. We have a number of the grapevines, the vigorous hybrids, they're asymptomatic. It really doesn't hurt them at all. So a lot of growers, if you have a block of a really nice hybrid that you want, you're reluctant to take it out. Rootstocks are also symptomless, so there are some problems. We do need effective control of vectors. That's required as well. So we move this buffer along, replant in behind it, do testing, and control the vectors. So testing is very important. It's going to be a major part of any control that we have. We're going to, whether it's roguing, whether it's these major pullout, we need a lot more and very effective and cost-effective testing of a large number of samples. So I just put this in here just to show you, remind you a lot of the work that Sud's been doing, perfecting the various test methods, PCR largely for grapevine red blotch. So fortunately, and hopefully you'll all hear about this soon, we're spearheaded here at CCOVI is an attempt to establish a platform for testing viruses. So different researchers, different areas can feed into that and have their material tested at a reasonable cost. That's a very important part of this whole program if we're trying to get on top of this virus program. So secondary spread of virus by insect vectors. So soft scale vectors of grapevine leaf roll disease. So leaf roll 1, leaf roll 3, they're both transmitted by a number of species of scale. The one I have highlighted in red are known to occur in Canada. So we have cottony maple scale, you have it here, European fruit licanium scale, very common soft scale pests in vineyards throughout most of Canada. They have a wide host range, they're on a lot of other plants, perennial woody plants as well. If you read the literature cottony vine scale and brown soft scale have also been reported from various areas in Canada. So we think we might have some of those, and I'm going to come back to that in a minute about what we're trying to do to find out whether we do or not. So we have a number of soft scale vectors of leaf roll in Canada. We also have a number of mealybug vectors of leaf roll. And again, in red are the known ones. Grape mealybug, it's known, again, wherever you grow grapes in Canada, you will have grape mealybug. Also, again, reports from the literature that we should have apple mealybug and Comstock mealybug as well. So it's possible that we actually have 3 species of mealybug vectors in Canada. So Sud's been working on this as well. He's very busy. So we've been collecting a lot of samples and barcoding them, checking with the database. So he's done this, and definitely you can see the matches here when you do a blast, the sequence data against what's available. And he's used a couple of different databases. So Pseudococcus maritimus, so that's the grape mealybug. And it's a very good match. We definitely have that. We have lots of numbers. That's our dominant species in British Columbia. We also have the European fruit Lacanium scale, Parthenolacanium corni. I always trouble with that one. Soft scale, and again, a very nice match. That's great. There is a problem though. The other species of soft scale possibly present in BC, the genetic sequences for the cottony maple scale and cottony vine scale, they're not available in the database, which is really surprising because these are major pests across Canada, not only on grapes, but on a wide range of plants. They're a big problem in nursery production of ornamentals, etc. So that's a little bit surprising. So we're going to be doing further morphological and molecular work to confirm what we actually have. So that's, and we'll get to the bottom of it, but he's doing a lot of work looking at sorting all of this out, looking at the genetic, using genetic methods. We also work with people in Ottawa to look at the taxonomic methods as well. It's important that we find out how many species we have in BC, what are the major vectors of the disease. This is a picture of how bad some situations can be. You can see these adult female grape mealybug here on this shoot. They're often towards the bottom. So we have the bottom of a shoot here. They'll often get on colonize the bottom of a grape cluster. So the bases of stems is really preferred. And they'll lay their egg clusters, these cottony masses here on the bark, on the more woody parts of the tissue. So you can see a cottony egg mass that contains the eggs there. So we have that one, and it's quite serious. Some numbers can get very high. We've been doing work looking at the generations or the population development of these two using sticky tape. You can put this out around the stems of the vines throughout the year. Go check them every week. And you can see from these results we have, as expected, 2 generations in BC. The biggest one happens early in the season. It's occurring now, where you get, this is the crawler stage, the first instar stage of grape mealybug. The reason that's important, the small instars, the larval instars, are more efficient vectors. So they've done the work, yes, they're more efficient. They're also the mobile stage. The crawler stage, they don't really even look like mealybugs, and they're very small. They can actually be blown on the wind. So that's the one we really focus on. That's the problem. Here's some pictures of the scale insects. So there's the typical picture taxonomically or morphologically. It looks like European fruit Lacanium scale, and yes, it matched up really nicely with the genetics. Cottony maple scale is a little bit different. Here's a picture that we're calling cottony maple scale, and this one really does look like it. And these masses, unlike mealybugs, for the cottony maple scale, this is all full of eggs that will hatch into the crawlers. So you can get instances where the whole trunk and the cordon can be covered with scale insects, the adult females with these large egg masses underneath the rear end of the female. So again, just to outline where we're at here, We do have the European fruit lecanium scale up here, verified. Yes, we've got it, not surprising. The problem is this pulvinaria group down here, cottony maple scale. They're the common one in the literature. We very possibly have cottony vine scale as well, maybe others. We sent some material a long time ago to Guelph, where they do sequencing, and came back with 5 genetic sequences, distinct genetic sequences. So we've got a little more work to do to sort that out. So lots of the disease, lots of the vectors. Do we have proof that it's actually being transmitted in vineyards? So here's the first one I'll show you. We have mapped a number of sites where we actually go out year by year, we follow the spread of the virus. So here's, we'll just call it Blockade. And you can see mapping over a 3-year period that there's been an increase in virus infections of 8.6%. So there's a block of Cab Franc, 500 vines in here. We go through and visually inspect them. On the reds, it's easy, and then you just go back and verify that yes, that is leaf roll virus. So it's spreading quite a bit. There's a vineyard adjacent to it with 100% leaf roll incidence, and you can see clearly that here's the edge, much higher. So that's come across the road into that block, okay? Here's another block, Block B we'll call it, of Merlot, 480 vines, and over that 3-year period, 18.4% increase. Here's another one. We went back, we started this one in 2011, and this actually, it's a prime block. This is their best grapes. This went into their premium wine. Virus had come across the road, had started on the edge. Most of the block was fine, free of disease. So we went into this area that where it had first come in, and you can see, it's hard to see these individual dots, but if you look over here, you can see that the increase over that period of time, 30% increase in virus infection over the 5 years. Okay, Block D. Here we are up in the North Okanagan. All of the previous ones were the Central or South Okanagan, and we had quite a significant rate of increase. I should also mention we did not see high populations of vectors in there. We were monitoring mealy-bug scale at the same time. They were actually quite low. We've had sites that have much higher populations. So that would be sort of a typical situation with quite good vector control. In the North Okanagan, where I mentioned, we have very few virus vectors for a number of reasons. Monitoring over 3 years in this Merlot block, 344 vines, there was absolutely no spread of the virus in that area. So this is where I was mentioning that roguing in some locations can work. You can go in here, very few vectors. The vines probably came in the nursery material. The infected vines came with it, and you could go in and rogue those out of there. And so some areas, it's very effective. So now we're moving on to red blotch virus. As I say, it's quite new, so there's very little known actually about the vectors. So there was secondary spread of red blotch virus in California, but the incidence was quite low. They were reporting an increase of 1% to 2% annually. Still, over time, that's going to build up to a significant amount of infected vines over time. In 2016, Batter et al. In California, under controlled lab conditions, showed the alfalfa tree hopper was a vector of red blotch virus. Now, we have tree hoppers in Canada. We have them in BC, you have them here. We don't know yet how many we have and how effective they are. There's likely some other ones that, some other leaf hoppers, especially the sharpshooters, that are likely to be vectors as well, and that's some of the work we hope to do. So grapevine red blotch virus, it's been identified, found in field collected insect species belonging to the a number of these homopteran, more like the tree hoppers, cicadelids and membracids, tree hoppers and plant hoppers. But more work needs to be done. If you detect virus from an insect, it doesn't mean they're a vector. It means that they have fed on that plant. It could simply be in the gut. It hasn't been cleared yet. Have to look at the details of this work. If you hold them for a certain amount of time and you test it from the vector, If it's gone, it's called a circulative persistent or semi-persistent type of transmission. So the vector actually gets infected with the virus. So the virus passes through the gut wall into the hemolymph, and then it moves into the salivary glands and then gets put back into the plant. So you get this circulative system. So there are various barriers. The virus needs to pass those to be before the vector it's a competent vector of the virus. So more work needs to be done. So far, we have one vineyard where we mapped only for 1 year, no sign of spread in BC. We've mapped a few more sites now, so we're hoping to find out if it's actually any spread in BC. And we're doing studies on the insect vector capabilities. Oh, I should go back though, just to show you. This is a little picture down here I got from the web of one of the tree hoppers. So this one is the three-cornered alfalfa tree hopper. So we have that one, another one called the buffalo tree hopper. As I mentioned, they are pests. Occasionally you'll get shoots on grapes that'll be flagged red, and you go look, you'll see that typical. I don't know if you can see it clearly, but you see this girdling of feeding down here, and they actually cause the death of it goes bright red. You'll see this red flagging in your vineyard. That's a sign of tree hopper feeding. So what do we do about this now? The important part, chemical control of grapevine vectors is important. Insecticides can be effective due to the prolonged feeding times required for virus acquisition and transmission. So as I mentioned, the viruses have to get into the insect, they have to get into the hemolymph, into the salivary gland, so they need to feed for a prolonged period of time. Feeding up to 24 hours is beneficial. You get higher rates of transmission, and they can retain that virus for quite a while. So insecticides can be effective for the control. One of the first strategies in this picture, we were running a trial just recently. We want to know, some growers are using dormant oil or lime sulfur for disease management, but it also will help control these vectors, the mealybug and scale. So we've got a plot with quite a number of these vectors, and we're out putting oil sprays on. We're combining this with sprays of some of the later foliar sprays as well to see what control we can get. So really starting off the season with dormant sprays of lime sulfur or oil against the overwintering eggs and nymphs would probably be beneficial. As I mentioned, the overwintering, you saw that larger peak on the curve of the crawler stage. That's early season. So if you can knock that back, that's probably going to be very helpful. Foliar sprays a little bit later when you've got a little bit of leaf material out, targeted for the crawlers and the nymphs that have been moving out onto the green material. These are the materials the insecticides that we have registered at the moment: insecticidal soap, Movento, Malathion, and Clutch. So these are all targeted for once they've settled down on the green tissue to feed. We have to do a little bit more on this. And we also have to look at what effect other sprays have on the vector population and the transmission of the virus. Because sprays for other grape pests can impact vector populations. So what we find in BC, large outbreaks of mealybug and scale recorded 1 to 2 years after a spray of a neonicotinoid spray. So things like a Clutch or a Sail or something that's one of the neonics that's been applied for leafhopper control typically. So what happens, we don't have to spray for many pests in BC. The neonics are very effective. They will give you effective control of mealybug and scale that first year. The second year, it still persists into the following couple of years, but it's not effective for mealybug and scale control, but it appears, we think, that it is still limiting the effectiveness of the parasites that help keep the mealybug and scale populations under control. So we're seeing quite a rebound, if you will, of mealybug and scale following the use of certain chemicals. And then there's also this situation where, summer oil sprays might be effective. They're not registered right now for mealybug and scale control, but we want to look at that. So the situation on Ontario vineyards is likely different. And the reason for that, different pest complex, different pesticide use, etc. So I would not want to take our situation in BC and extrapolate to Ontario. That work will have to be done here to find out if you're getting these resurgences of this mealybug and scale and what your best approach might be. We do need alternative, effective, and selective spray materials. They would really benefit the program. In summary, we've shown a significant increase in the amount of grapevine leaf roll virus since the previous survey in 1996, a very low incidence of grapevine red blotch in BC vineyards. Leaf roll 3 is the predominant virus. It's widespread in certain areas of the South Okanagan, where we've shown with mapping studies that a high yearly rate of increase. The vectors of red blotch virus are recorded, but no documented spread in BC as of yet, but we're going to follow up on that. We have at least 3 species of leaf roll vectors in BC vineyards, possibly as many as 5. The populations vary by region. The effective virus management requires, first of all, tech transfer. I haven't really gone into this a lot, but we need to get a lot of information out to the industry about what the problem is. This wasn't even really recognized until quite recently. So it's a serious problem now. We need to get the information out about what we found and how to manage it. We need virus-free planting material, we need roguing, we need vector management. Of course, we also need quite a bit of virus testing as well. In some areas, a coordinated area-wide approach would be of benefit where virus and vector levels are very high. And with that, I thank you.
There's some questions. I have a question now, Tom. With our changing climate, even if I'm thinking about this in terms of what's happening between North Okanagan and the South Okanagan Valley. Is part of, might that change with a warmer climate? What's causing those differences?
Well, the difference between the North Okanagan and South Okanagan, climate could play a role. We get quite a difference in heat units between the north and the south. But there's also a big difference between the soil types. The South Okanagan is very sandy. Some of it's just like deep beach sand. And as I mentioned, some of the ant species really thrive there. So you get a lot of different ant species, large numbers of ants. The sprays are very different as well. The size of the vineyards in the North Okanagan tend to be smaller, and they're often surrounded with either native vegetation or orchards or some other situation. So it's contiguous in the South Okanagan where you have plantings adjacent to one another. So there are a number of things. Climate change could play a role. All of these insects do have favorable conditions. And for example, mealybug, they're more of a subtropical group of insects. So if you go down to Southern California, they have, I think now, about 8 or 9 species of mealybug they're dealing with. So far we have 1. We know the apple mealybug is reported. We know we should have it. We just haven't found it on grapes yet. And it could have been a mistake. Somebody saw grape mealybug. They're quite distinctive though. You squish a grape mealybug and it's kind of an orangey fluid comes out of them, the hemo. And the other ones, it's a grayish, clear. So I've squished a lot too and haven't found anything other than grape mealybug. So the warming conditions, we might see a bit of a shift where you'll get more mealybug. We really don't find any mealybugs in the North Okanagan, just the soft scale. And there's more of the mealybug problem in the south. So there probably is a component of that. Linda.
Hi, Tom. I just wondered, you mentioned earlier on that the young vines, you get a lot of vines in, bring them in from California.
Yeah.
And that's why you're looking at the younger vines to have to look at
for the red blotch.
What percentage would you say, you might not have these exact figures, but what percentage of vines are brought in from Canada, from California, what percentage from other regions, how many what percentage is already cultivated?
Yeah. I'm not the one to ask, and it would actually take, Deb probably knows better than I do. For BC, there's also been a shift we used to get the bulk of our material. Now there is some domestic production, so Ontario has some and other regions have some. We used to get the bulk of our material from France, and actually a lot of the rootstocks from Germany, and they would be produced there and shipped here. For various reasons, largely because the CFI delisted a lot of nurseries in France a few years ago, there was a big concern about phytoplasmas. So this is another group of diseases that were getting into a lot of the nursery, the big nursery operations, and CFI delisted a whole bunch of them. So that was one of the concerns. Also, when they were deregulated, a lot of the material did have leaf roll virus and other viruses in it. So our industry, and I don't know what happened in Ontario, but they started getting more of their material from Washington State and California. There's concerns with that too, why they didn't do that previously. They're not as concerned. Some other problems that the crown gall in particular in California, they don't have the problem because the weather conditions don't, it's not expressed. So you take the material has crown gall, you move it to BC or Ontario, you get a big problem. So they preferred French or other material. So there's been a big shift in where we get the bulk of our material. And I've kind of dodged the issue. The BC Wine Institute and maybe the Ontario Grape and Wine, they could probably give you a bit better. But there's also some mixing too. When it's a difficult thing to track because you buy the vines, you place an order with, there's brokers and then where are they getting things from, so.
Yeah.
Yes?
I spent the spring and summer counting mealybugs.
Oh.
I was just curious, are you finding most of your mealybugs off the trunk and up into the actual stem of the canopies, and that's where you're finding them?
Most of the mealybugs, as I showed in the picture there, they really like to be at the base of the green tissue for the current year. So if you want to find them, you hunt at the base. For the winter, as I showed the overwintering, the egg masses, they will be on the woody tissue. You have to remember too, though, that the crawler stage and the small nymphs, they move out onto the leaves. How we find them, the best way that we can find them, if you look for the ants at that time when the first few leaves are out, the ants are they'll be scurrying up and down the trunk and looking, and when you first go out, they're clear. You can't really even see them in the field. You bring them in under the scope and there's all these little nymphs settle down on the leaf material. So it's a progression. The crawlers go out onto the leaves and then when they're bigger, they move to the base of the stem or the green tissue. And then later on, they move to the woody tissue to lay their eggs. So you kind of have to follow them through. Where did you do your sampling, by the way?
Here.
Here?
Yeah.
So this is all new in Ontario. Yeah. Well, they're as I say, if you look, they're mainly right at the union of where the new current year's tissue joins the trunk, the woody tissue.
Question. I just want to confirm what you're saying early in the season, like even when it's just starting to grow, like as soon as tissues is emerging?
Yeah, as soon as tissues are emerging, so the crawlers will be moving out. As soon as they can get some green tissue, they'll be feeding on that. So these little tiny little specks, and they're a pain. We have these crew of people with this clear sticky tape, and there's debris and there's all kinds of things on that. They're looking for these little specks on there. I don't know if you did any of the tape work. And then they'll move out as the leaves expand more throughout the year. They'll be on the leaves as small nymphs, and then when they're bigger, they'll move to the base of the stem.
So I think there's like hormones drug for the males personally. Have you been able to?
We're going to be doing that. Wendy tried it. They're kind of interesting. We're going to try it here. In some regions, the males are supposedly quite common, but a lot of the Homoptera have flexible reproductive strategies. And so some of them, they can reproduce parthenogenetically, so that asexually, so they don't produce males and they just carry on. And in the northern climate, maybe we don't see a lot of males, so I'm not sure that we really have that, but we're gonna put pheromones out and see what we can find.
Yes, so we have a question from someone watching online in
Sure.
Canada. She was wondering if we could elaborate a little bit more on the movement of infected bees on equipment. Essentially, she's asking if the infected bees themselves can spread the virus onto another vine. Just kind of
Yeah. It's a good question because no, it doesn't get spread. None of these are known to be spread mechanically. So if you go through, it's not the leaves themselves, but if those leaves are carrying vectors. And so if it's material that's infected, so the leaf is infected and it's carrying vectors on it, and those vectors, as the leaf dries up, they're going to crawl off onto something else. So it's not the plant material itself. There's no mechanical transmission known for these. It's all through these vectors.
Well, it's much clearer to me that
Well, Tom, thank you so much for a very informative talk today. And here's a small token of our appreciation for coming and visiting us and sharing your knowledge. We have a lot of work to do here in Ontario, but we're very excited about looking at our results in the context of what you've already shown in BC.
And thank you for having me. And that's a wonderful way to close, is that we will be collaborating quite closely with Ontario. So we have a very active, great group out there, and you're forming some people to do some wonderful research out here. So there'll be ongoing, lots of close collaboration between the two groups, and we look forward to that. So thank you very much.
Thank you. And I would just like to thank everybody for attending our lecture series this year. It's been a great number of talks that we've heard. Please remember that if you missed any of the talks, you can always go online to our COVI website and look under our Outreach Services tab. That will bring you to the COVI Lecture Series, and you can always watch any of the lectures on our YouTube videos that are posted. And continue to watch us for updates. Come back to our website often, and we'll be sure to get the word out for next year's lecture series that will start in January of 2018. So thank you very much for your time.